This session is focusing on precision medicine issues. These came up earlier in the day, and this session we're going to focus more specifically on such issues as trial enrichment, biomarker, qualification in science, and mechanistic reasoning to optimize patient selection. So a host of issues, but again, touching on topics that we've already been discussing today. These are approaches to improve patient selection and trial enrichment, to establish proof of concept or to enhance dose finding, and also to define parameters for translating early stage or early phase biomarker discovery to confirmatory trial designs. As with our other sessions, we're going to start off with some lead presentations, then case studies, and then further discussion, hopefully including many of you. We're going to kick off with a presentation from Michael Pakanowski, who's the Associate Director for Genomics and Targeted Therapy in the Office of Clinical Pharm at CDR. We're then going to discuss and explore biomarker strategies in more depth with case studies from Vicia Viglietta, the Director of Medical Research at Biogen, and Jim Sullivan, the Director of Clinical Biomarkers for Vertex. And after the case studies, we're going to hear perspectives from Rich Mosiski, the Deputy Director for Science Operations at CDR, and Alice Chen, the Head of Early Clinical Trials Development Program in Division of Cancer Treatment and Diagnostics at NCI, and Stephen Friend, the President and Co-Founder of Sage Bionetworks, among other things. So look forward to, again, a rich set of presentations, and Michael, I'll turn it over to you.
Thanks. I'd like to, special thanks to Brookings for organizing this meeting. I think it's great to get all of these folks together, and certainly takes a tremendous amount of work to get it together. And also to the panelists for taking time out of your schedules to be here today. I've been advised that some of you may have had large lunches, so please, I implore the panel to be very active and provocative to keep us awake here. So what I'll talk about for the next few minutes is precision medicine aspects of early stage drug development. So this is somewhat at odds with some of the topics we were talking about earlier, where there is an interest in getting a diverse array of patients into early phase studies to allow us to have some variability built into the clinical trials to explore dose and exposure response relationships. So we're going to look at this a little bit from the other end of the spectrum and how we can actually target these early trials a little bit more precisely to maybe perhaps expedite establishing proof of concept in some of these other steps to get to phase 3. So over the course of this session, we'll try to identify some design practices at the trial level, but also at the program level that will enable these late phase enrichment strategies, hopefully which will reduce attrition and improve individual patient outcomes, obviously. So for the purposes of our discussion today, when we're using the term precision medicines, what we're really talking about is drugs or biologics that are intended for use only in a subset of patients who are identified by some genomic, proteomic, or other biomarker. This is not to say that there aren't myriad ways to personalize therapy. We heard about many before, using clinical factors such as age and sex to personalize the dosing. Clearly, that would also fall under this umbrella, but for the purposes of our discussion, we're really talking about molecular profiling and biomarkers. Now, the biomarkers being used could have some diagnostic value, predictive value, or some other value in informing what the disease state is in a patient. And one thing that, from a clinical pharmacology perspective, we're obviously interested in is the key assumption that there is some mechanistic relationship between the biomarker and the activity of the drug. And that really leads us to this putative predictive utility of the biomarker for predicting treatment outcomes. Now, to get to the end result of having a precision medicine, clearly there are many pathways to get there. In some cases, it may begin very early in the course of development, stemming from a well-understood mechanistic relationship and a very well-understood biomarker. But at the other end of the spectrum, we may have experimental evidence or clinical trial subset analyses that might inform whether a precision strategy is pursued. Now, the reason we're here today is because there is quite a bit of activity in this area. As you all probably well know, we've approved a number of drugs with companion diagnostics over the past couple of years. There's certainly intense interest in this in the setting of oncology, perhaps less so outside. But just to give you a sort of sense of what the pipeline is looking like, if we went and looked over upwards of 2,000 submissions over the past year to look at the content and nature of the questions and meeting packages and what the trial design characteristics look like. We estimate that about one in five packages has some biomarker-related component to it. Now, this could be an endpoint or enrichment factors or patient stratification, and that's spread across all different phases of development, but clearly there's quite a bit of activity in this area. So something we need to pay close attention to and begin to develop an understanding of how to make decisions around. As would be expected, a lot of this activity is in the hematology and oncology area and peppered around the other therapeutic areas to a lesser extent. So clearly there's a lot of activity in this area. Now, foremost among the decision to pursue a targeted strategy is having the right candidate product to do that. Now, it's pretty clear that if you have a drug with high variability and a steep exposure-response relationship, you do want something to help optimize how you dose patients in the clinic. It's important that you don't tip patients over that edge of toxicity or put them in a range of exposures that are sub-efficacious. But additionally, there may be other areas where a personalized strategy is attractive. This could be in the setting of morbid diseases that you can't monitor in the clinic to predict what an individual outcome would be, and also for drugs that have less than favorable toxicity profiles to help shift that risk-benefit. More obviously, if a drug is subject to polymorphic metabolism or the drug target is clearly variable across individuals, that would also lend itself well to a precision strategy. Now, putting this into the drug development context, we obviously have a phase of exploration in drug development and then a phase of confirmation of drug effects and clinical benefit. So in the exploratory phases, there's a couple of different approaches. You can either take the all-comer approach, where you're generating information and looking to explore and identify novel biomarkers that might predict the patient's response. These could be susceptibility, prognosis, prediction, or response-based. And then on the other hand, you might actually know something about a biomarker or the drug and use that information to help streamline those early clinical trials to help give you the earliest read and proof of concept. Once you've got that information in place, you then pursue to later phase trials where you're confirming clinical benefit, and the basic maneuvers here would be either enriching the clinical trial population or stratifying randomization to test different hypotheses. The ultimate end game would be, using in the clinic some test that would help you pick the dose, select which patients get the drug, or help you monitor the patient. Now, I think in late phase confirmatory trials, a lot of the problems have been discussed ad nauseum. There's a lot of issues with who or when and why to enroll marker-negative patients in later phase trials to understand the merits and limitations of a biomarker, how to construct the hypothesis testing strategy, how you optimize cut points, and how you simultaneously prepare for and perhaps prevent the need for a companion diagnostic that's ready to market once you get the drug approved. So those issues have been sorted out quite a bit. If we're shifting a little bit earlier, there's obviously a number of other issues that we have to contend with. In early phase trials, we're dealing with a relatively data-poor environment. There's small sample sizes often, which really limits your ability to do a lot of these exploratory studies to identify biomarkers that would predict response. So we have to deal with issues such as multiplicity. Using high-throughput omics technologies obviously can lead to a lot of false positives. Additionally, early phase studies aren't necessarily designed based on clinical endpoints, which may lead to some uncertainty as to how a drug and biomarker relationship would translate to the clinic. Additionally, the analytical methods may not be very well set up at this stage of development. You may not have a really proper functioning assay at this stage. And all of these things can lend themselves to false conclusions and potentially undermine the pursuit of these types of strategies. So, the ideal scenario, co-development of a drug and a test is really planned from the outset. However, as we well know, this is really suitable only for those cases where we have clear predictive and prognostic biomarkers that have been already well established, or there's some clear difference in the drug target. For instance, some individual patients don't express it at all. Now, some could argue that you could take the alternate strategy and even in the absence of information about the relevance of a biomarker, you could still streamline that early phase of development to select only certain patients to help minimize the amount of noise in those early phase trials, and then scale up as you go on into later stages. This is certainly rational, but not something that's necessarily commonplace at this point. More typically, what we see is exploration in phase 2 trials coming to the agency with proposals to streamline the phase 3 development with the use of these types of patient selection biomarkers. Now, I mentioned there are several limitations to this approach related to sample sizes and the amount of data you have, but there are some advantages because you are really set up in that context to only find biomarkers that are really likely to be clinically relevant, just because it has to have a very large effect by nature of the study. Additionally, you also do gain some evidence of predictive value because you have patients who are marker positive and marker negative, which then can help inform how to design the later phase studies. In some rare cases, we have had some biomarkers that have been identified in late stages of development or even in the post-approval setting. In these situations, there's really some compelling shift in the risk-benefit relationship that would necessitate use of that biomarker after a drug has already been in use in a broad patient population. So in thinking about whether a biomarker-based strategy is appropriate, it's also relevant to consider what the long-term outcome might be once an NDA might be filed. And looking back, just based on experience of the drugs that have been approved with companion diagnostics or other biomarker tests, it's clear that there's a couple of different factors that weigh into that decision-making, and it is still fairly nuanced. But obviously things like the nature of the biomarker, if medically relevant biomarkers that are used in clinic translate very easily to this precision type of strategy, whereas novel biomarkers do take a lot more learning in the early stages of development. The nature of the disease is also quite relevant in some cases, assuming you don't have toxicity issues, but when there's no available therapies, it might be desirable to give broad access to a drug without using some patient selection biomarker. Conversely, if you have a drug that has an unfavorable toxicity profile, it may be necessary to include some type of testing strategy in the clinic to help offset that risk and shift the risk-benefit profile. So those are the considerations at the later stages of development. Now, if we take all of this just to sort of put it into sort of the broad drug development context, this is sort of the idealized development scenario. So you run your early phase studies, and you're looking at mass balance and pharmacokinetics and safety in the very early stages of development. There's obviously not too much time and opportunity to learn about determinants of drug exposure in this context, at least from a genetic perspective. But that doesn't mean that over the course of an entire program, because phase 1 studies do occur, obviously, across all stages of pre-market development, there is opportunity to use information from bank samples to learn what might be determinants of drug exposure variability. On the flip side, you may also be able to apply some information in the early phases of development. So to the extent that you're enrolling patients in your early studies, to get that patient population homogenized to a point where you can establish proof of concept and really get a clear read on the pharmacokinetics could be desirable. Once you get into phase 2 studies where you're exploring more of the exposure-response relationships, the safety profile and efficacy profile, this is when you start to have opportunity to learn a bit more about the determinants of response variability from a genomic perspective or proteomic or what other perspective. And additionally, we also have that opportunity to enrich to help streamline that demonstration of clinical benefit. But this is also a time, when you're running omics studies and these early phase studies, you can learn. If you have more than one phase 2 study that's being conducted, you do have some opportunity built in to confirm whether or not a biomarker drug response relationship does exist. And this really is what serves to set up the design of phase 3 trials so that it's executed with the highest probability of success. Now, once you get into phase 3, there's a number of different approaches. You can enrich. If you're doing 2 phase 3 trials, you can enrich one and potentially have an unselected population for the other, so you still get to learn about the biomarker in that context. So there's a number of different approaches to carry this out in phase 3, including some strategies such as fallback hypothesis testing and other ways to evaluate the effect of the biomarker. And clearly, the question's often been raised as to when you can learn about biomarkers in the post-marketing setting. And so in the interest of streamlining development, enrolling only marker-positive patients in the early phases and even confirmatory trials, there may be room to actually conduct these studies in the post-marketing setting, although those cases have been quite exceptional up to this point. So just some points to consider from my personal perspective. I think we everyone involved in the drug development space really needs to manage very proactively variability in exposure and responses. We need to take advantage of known biology to achieve this sort of quick win, fast fail type of model of drug development, which may prove successful. And we need to, I think, design trials that are really fit to identify these predictive biomarkers. So that, again, goes to the theme of today, I think, which is investment in phase 2, getting a large sample size, dense information about each individual patient to help do the most learning that we can. Clearly, there's a number of challenges that's not practical at this point in time. We really have to figure out how to build confidence with very limited data in small patient populations, how we develop around known liabilities, such as polymorphic metabolism or QT prolongation. There certainly may be precision strategies to help achieve that. Generating information in a drug development context can be a scary thing. You can't unsee things. So there is an issue related to generating unexpected findings and how that will affect the course of a program while still trying to do this exploration and learning about the molecule. Companion diagnostic co-development is clearly an issue that raises many challenges because you're developing 2 products simultaneously. There's often been calls to have discussions with the FDA very early in the course of assay development, so you're set up to have a marketable test at the end of the development program. But those pathways do have to be aligned from a timeline standpoint. And then, there's also uncertainty at this point about how to integrate hypotheses into late phase trials. There's no set approach to doing fallback hypothesis testing where you still retain that opportunity to win in the entire population, even if you go in with a biomarker-based hypothesis for a narrow clinical benefit. So I think that those are some areas where continued work might be good. Hopefully, over the course of this meeting, we'll hear some additional challenges for FDA to consider or potentially some solutions to some of these issues. With that, I'll close. Thank you.
Michael, thanks for that overview, and on to Visya for some of our first case studies.
Good afternoon. I would like to share with you today some of the results of the phase 1b interim analysis with Aducanumab, which is a monoclonal antibody against amyloid, in patients with prodromal and mild Alzheimer's disease. I don't think I need to remind you how devastating this disease is, and the numbers are quite frightening. 60 to 70% of the dementia diagnosed today is due to Alzheimer's disease. Only in the US, we have 5 million people with this devastating disease, and the estimate is to rise to 16 million by 2050. Obviously, this puts a lot of burden on families, loved ones, and an economic burden on government and society, because to take care of these patients, we spend huge amount of money, 16 billion globally, just for the health care of these patients. What is it, though? What are the features of this disease? 2 main ones that we have known for decades: the accumulation of amyloid beta plaques in the brain and the accumulation of neurofibrillary tangles, and you can see them here. All this, together with maybe the contribution of inflammatory process, will lead to brain atrophy. You can see how the brain of this patient shrinks on the right-hand side. And ultimately, patients will lose their neurons. And the symptoms relentlessly will continue on, and they will lose clinical and functional aspects until death. 8 years from diagnosis, these patients will be dead. So the amyloid hypothesis is something that has been known for decades now. The question is, why don't we have a drug yet, right? So we know that there are A-beta peptides that are produced and are forming aggregates both in the soluble space and in the brain itself. So there is a continuous enzymatic cleavage of the amyloid precursor protein that will give birth to peptides that can accumulate. They are toxic, and they can accumulate in the brain. So why don't we have a drug? We have a target, right? So it has been known for years now that accumulate accumulation of amyloid plaque in the brain is the hallmark of Alzheimer's disease. And the trials that have been run so far maybe have had some deficiencies. First of all, many of the trials didn't really enroll Alzheimer's patients. Retrospectively, we have learned that up to 36% of these patients didn't have amyloid in their brain when they were treated with an anti-amyloid agent. So maybe that's one of the causes why the drugs did not work. We didn't enroll the right patients. Also, we enroll patients at the later stages of the disease when drugs might not be no longer effective. So the other reasons, maybe we didn't find the right doses because we were scared about adverse effect that anti-amyloid agents might cause in these patients, namely ARIA. It's an imaging abnormality due to removal of amyloid in the brain that causes kind of swelling of the brain. So we didn't have the right population, we didn't have the right dose, we have patients at the wrong stages of their disease. So I think, thankfully, we are learning from our own mistakes, and hopefully we are doing something differently these days. So I want to show you the case of Aducanumab, which is a human monoclonal antibody that is selective for aggregated forms of beta amyloid. So it selectively binds to the amyloid that is accumulated in the brain, not to the monomer that are in the periphery, but the plaques that are in the brain. We have had some evidence of efficacy in a mouse model where aducanumab was able to remove plaques from these mice in a microglia-mediated phagocytosis manner. We did single ascending dose studies to demonstrate safety and tolerability of each one of these doses up to 30 milligrams, and then we moved into a phase 1b study that we keep calling phase 1b, but it resembles more a phase 2 studies because it's very large. It almost enrolled 200 patients. It's a dose escalation study, a dose finding study. But what did we do that was different from other studies? First of all, we enroll a population early on in the course of the disease. Only prodromal and very mild patients with AD were enrolled in this study. The second feature that is quite crucial is that every patient underwent amyloid PET scan to ascertain the presence of amyloid in their brain. We didn't use any other methodology but imaging to ascertain presence of amyloid. We did test several doses from 1 to 3 milligram, then 10 milligram. Then we were supposed to go even higher to 30 milligram, but we have realized that the incidence of this adverse event was too high, so we backed down to 6 milligram to have an intermediate dose between 3 and 10. And then also, in order to reduce the occurrence and maybe the severity of this adverse event, we included later on a titration regimen so that the amyloid could have been removed more slowly and maybe prevent the incidence of ARIA cases. Obviously, this was designed as a safety and tolerability study, but it got offense, I guess, because we have a number of pharmacodynamic and clinical endpoints that we evaluated throughout the course of the study. This is just to give you an idea on the disposition of the patients and the baseline characteristics. We had both patients with ApoE4 carrier and non-carrier. Now, ApoE4 carrier status has been also known for many years as being really related to AD, and we see here that the 2-to-1 composition of this population, so 2 carriers, 1 non-carrier. So, and also we had a split between prodromal and mild, with the vast majority of the patients being mild. And those are the characteristics of clinical characteristics at baseline, very equally distributed across the treatment arms, and the presence of amyloid. Here is the quantification of the amyloid in the brain, around 1.4. Results, what did we see? At 6 months, we saw a dose-dependent reduction of amyloid in the brain of these patients with high statistical significance at 3, 6, and 10 milligrams, and also was time-dependent because the reduction continues over a year at the end of the study, again with high statistical significance. We also took a peek at the clinical assessment. This is a clinical dementia rating scales, and looking at sum of boxes. In the first 6 months, nothing happened, so there's no separation between placebo and active arms, but continuing, we see the yellow line is the placebo continue to decline, at least they lose 2 points, whereas there is a dose-dependent treatment effect at all doses, reaching statistical significance at the highest dose, 10 milligram. The second clinical endpoint that we look at is the mini-mental state. And again, we didn't see any effect at 6 months, but the placebo, yellow line, continues to decline at least 3 points, but we saw a dose-dependent treatment effect of every one of the doses. And there is a dose response overall that is statistically significant at 1 year. So we have seen those imaging abnormalities, and they seem to be ApoE carrier and dose dependent, with the highest incidence in the carrier patients carrying this gene at the highest dose. So in summary, what we have done is demonstrated a time and dose dependent reduction of amyloid plaque by looking at PET imaging. We have looked at a significant effect on clinical parameters, and we have also evaluated ARIA, which is this image abnormalities that is dose and ApoE carrier dependent. But it seems to be monitorable because by doing frequent MRI in the patients early on, we can prevent further problems. This led us to develop a phase 3 study, 2 actually phase 3 studies that we have just started in June, and the learning were going early. We are looking at mild cognitive impairment patients due to AD and a subset of mild AD. We are still using amyloid PET for enrichment of this population in all subjects, and you can only imagine the feasibility nightmare that is, because the PET availability, PET is not available in every geography, and the tracer are not really available in every geography. We are also looking at treating patients differentially according to their ApoE4 status. So we have introduced a titration regimen thinking that the safety profile can improve, and we have established 2 doses, low and high dose, in each one of these subgroups. We could model also the duration of the study, since the results that we have seen at 1 year with fixed dose in the phase 1b mimic what we might be seeing at 18 months with a titration regimen. And the endpoints will remain the same. CDR sum of boxes will be our endpoint, and a number of clinical and radiological endpoint will be included. So I hope I showed you how a small phase 1b study led us to very solid results that made us design the phase 3 studies, possibly in a rational manner and promising manner for our patients. Thank you.
Thanks very much, Lucia, for that interesting presentation. And next is Jim Sullivan from Vertex.
Should I escape or just advance now? Okay. Okay, so I'd like to make 2 disclosures before I begin. First one is that I work for Vertex, and the second one is that I'm actually not a pharmacologist. And so in light of that second disclosure in particular, I want to thank the organizers of the conference for letting me come here and talk to you today a little bit about some retrospective summaries of what we've done in the space of CF development at Vertex. So in order to do that, we're going to begin with some very brief background, enough to put us all on the same page related to the disease pathophysiology and how the molecular determinants of that disease pathophysiology drove different development plans for different types of molecular defects, which we'll discuss. So very, very briefly, by way of background, as I think most people here are aware, CF is a life-limiting rare genetic disease. And because of the underlying protein in which the genetic defect causes The disease is expressed systemically. There are multiple manifestations of disease. The one which results in currently which results in the highest levels of mortality is the pulmonary manifestation of disease. And I kind of want to comment on a couple of things here just very briefly. The first one is that CF is indeed monogenic. It's caused by mutations within the CFTR, cystic fibrosis transmembrane regulator. It's an ion channel. It's an ABC transporter. It's located in the membranes of epithelial cells. But in spite of the fact that it's a monogenic disease, there are over 2,000 variants that have been identified, or approximately 2,000 variants that have been identified within the CFTR locus. And of those, about 143 have known associations with disease. That also means that there are a lot of unexplored mutations, and it of course means that there's a lot of variation in the ultimate consequences of disease at the molecular level. The other thing that I want to point out is at the bottom here, and this is something that really I just wanted to highlight given the conversation that happened during session 1, and it really began with Dr. Woodcock's remarks at the very beginning of the day, which is how clinical validation and utilization of natural experiments can help to inform clinical development plans. And I want to point that out in particular because of the fact that the association between the CFTR locus and disease was first published in 1989. So that means both that it took 23 years from the identification of the causative locus to the approval of the first compound that specifically targeted the underlying cause of disease, but also because of the fact that the clinical program benefited from an extensive amount of research that took place within a well-motivated patient community. And since I feel like I've spent half my time on my first slide, I'm going to move faster now. So we've already talked about this, so I'm going to cruise through it. But ultimately, cystic fibrosis is caused by a loss of ion transport, and that loss can be caused by either a defect in the quantity or function of protein at the cell surface. And ultimately, this loss of chloride transport results in multiple clinical manifestations and really what we can think of as a pathophysiological cascade, that leads to a number of different manifestations of disease that are pictured in this little caricature in the bottom. I mentioned already that the pulmonary manifestation is the one that's the leading cause of mortality in the current state-of-the-art of treatment. And it's also what is utilized for our indications, our clinical endpoints. Surrogates of those clinical endpoints are ultimately what allowed for registration studies. But I also want to point out that CFTR is expressed in the sweat glands, where it plays a resorptive role. And because of that, it allows for a stable manifestation of the intrinsic level of defect that's present within the CFTR protein within a patient. And one benefit of that from the standpoint of a biomarker and thinking about proof of concept biomarkers is that this is actually a clinical diagnostic biomarker. So it's already been utilized within the community in order to diagnose CF. And it also allows for relatively near-term readouts of the degree of treatment efficacy, not from the standpoint of clinical benefit, but from the standpoint of target modulation. When we look at that, the other thing that becomes immediately apparent as we look across the spectrum of different mutations is the fact that the disease is actually remarkably heterogeneous, and that heterogeneity is caused by the variable mutations that lead to the ultimate disease. As we look along here and we correlate the degree of CFTR activity against sweat chloride, which is that biomarker, that mechanistic biomarker that we can point to, we end up going from a situation where you have very severe disease that happens early on down to the carrier state. And this allelic series helps to inform the ultimate development path. So when we think about that development path and we think about the fact that the total chloride transport is going to result from the product of the amount of CFTR that's present and the functionality of whatever CFTR is present, and we recognize the fact that those 143 known mutations that are associated with disease fall into multiple classes, where you can end up with a gating defect. An example of this is the G551D mutation, which was the first indication for Kalydeco. Or you can end up with the most common mutation, which is the Delta 508 mutation, basically a deletion of codon 508 within the CFTR protein. And essentially what this results in is a trafficking or processing defect with a decrease in the quantity of CFTR at the cell surface. Ultimately, when we're utilizing phenotypic screens in order to identify scaffolds or molecules that have the potential of benefiting patients, there may be different classes of molecules that have the potential of benefiting a patient that carries a gating defect versus a patient that carries a processing defect. Ivacaftor is a CFTR potentiator, whereby it will increase the open probability of that channel, and therefore a gating defect, so a protein that is a decreased open probability, would it be expected to have a clinical benefit from administration. Conversely, a protein defect like the Delta 508 variant would be expected to benefit initially from a compound which is increasing the amount of protein at the cell surface, and then you would expect to see additional benefit by further increasing the open probability of the molecule once it's made it to the surface. So we're going to very briefly go over the gating defects. So those are the two main classes that we're going to talk about. One is the processing, the Delta 508, and the second are gating defects. So these are severe, the 10 gating defects that have been assessed clinically are severe mutations and ultimately have very little functional CFTR at the cell surface. There's plenty of protein, but that protein doesn't exist in a very functional state. This can be measured via single-channel electrophysiology measurements, wherein we have a normal cell type displayed in the left-hand column, looking at the degree of open probability over time. And when we look at these severe gating defects, you can see that there's a significant loss of open probability relative to the wild type. These assays are utilizing site-directed mutants and Fisher rat thyroid cells in order to evaluate then what will happen when we add our potentiator. And when we add Ivacaftor, which is indicated by the full bars, you can see an increase in the open probability regardless of the individual mutation that was assessed. And ultimately, these data then allow for assessment of that comparable marker of thinking about how it is that we're affecting chloride transport in vivo. And in that assessment, if we look at our phase 3 studies of G551D mutations indicated in blue and patients with other gating mutations indicated in green, you can see statistically significant improvements in both the degree of sweat chloride and also in the change in FEV1. FEV1 is a measure of spirometry, so it's a surrogate for the clinical endpoint that leads towards registration. So now very briefly, with our last couple of minutes, we're going to move on to talk about the Delta 508 mutation. As we said, this is different than the gating defects in that very little protein is able to make it to the cell surface because of the processing defect caused by the Delta 508 mutation. Ultimately, we're going to compare everything against the Delta 508 state. In contrast to the previous experiments, this is actually looking at a in vitro system that's derived from donor bronchi. So this is human bronchial epithelial cell assessments. And what we see is that when we add in Lumacaftor to the system, which is VX809, we have an increase in the quantity of CFTR at the cell membrane, which leads to an increase in chloride transport. What we then also note, given the increased quantity of CFTR that's present, if we add in the potentiator on top of that, that then allows for a further increase, which led to the decision to move forward with treatment with this particular patient population with the combination of a molecule that increases the quantity of CFTR and also one that will increase the open probability, assessing those in combination. These were assessed with 2 different phase 3 regimens, and the structure conducted in 2 separate trials was a 6-month placebo-controlled phase followed by an open-label phase in which the patients in the placebo arm continued on in the open-label period. And ultimately, overall, we have to talk about safety first. Treatment with both doses was generally well tolerated. In the active group, there was a higher incidence of dyspnea, respiration abnormal, flatulence, and rash. And the frequency of LFT elevations was similar between placebo and active groups. There was a greater number of SAEs related to abnormal liver tests in the active. And now if we look at, ultimately, again, the surrogate for the clinical endpoint of FEV1, and we look first at the placebo-controlled phase, where we have patients being treated with either placebo or one of the 2 active groups over a 6-month period, there was a statistically significant improvement within both treatment arms. And then that improvement was sustained throughout the open label period, which also included a significant improvement amongst those patients that had been in the placebo group. Thank you.
All right. Jim, thank you. I'll turn next to Rich. You can stay there, absolutely.
Since I'm providing a perspective, that's a very different ballgame. I can just talk from here. Well, the good news about talking at this stage of the game is that everything I was going to say has already been said. So in providing that perspective, I can at least try to underline some of the things that I think might be important, or maybe expand just a little bit on them. So precision medicine, there's no question that that's going to be very important for all of us in the future. And it has this symbiosis, this very clear relationship with biomarkers, if you already picked up from all of the talks that have preceded me. But I think it's worth taking a moment to sort of then put biomarkers in some perspective as well. So biomarkers clearly have many different uses in clinical research. In fact, so much so that I think we really need to, and we are developing a taxonomy for these biomarkers so that we don't get too confused in sort of interchanging the names around what we really use biomarkers for. So, and they can be used through the various different stages. Certainly in the early stages, they have a great utility, and then that translates into their use in the later stages. But I might even argue that they go way back to before the phase 1, 2, 3 paradigm that Mike was mentioning and others have been speaking to. There's even the phase 0 utility of biomarkers. Now, that may not necessarily be part of precision medicine. It may not necessarily be part of a regulatory thought process, but it can be key to how we invest in the future for where we think we should be developing products. And that's in this idea of phase 0 proof of concept, or the human experiment, if you will. Other people might call it experimental medicine, where you can use a biomarker that can be a pharmacodynamic readout of that drug effect, usually on the intended target itself, so that you really have some human confirmation that the drug in the human is going to do what you can really hope it will do. Ellie Gustat was mentioned previously, and I think back to when I was at Genzyme, and I've been on the biopharmaceutical side of this for more than 20 years. One of the things that really gave us confidence at the time that I was there was that we were able to show a very dose-dependent decrease in a glycolipid called GL1 that's in the pathway that this drug was designed to interfere with, glucocerebral ceramide synthetase. And so that beautifully was able to tell us that we really could do what we wanted to do, and that this was worth pursuing at the time when the company already had a treatment for that same disorder. But this demonstrated that we could do better with possibly going down this route. The other area from that time even goes back 20 years when we were looking at gene therapy for cystic fibrosis. So in this case, we were trying to insert the CFTR gene, but that hadn't really been done in humans. It had been done in animals, it had been done in vitro cell cultures. So we were trying to lay the viral vector with the gene onto the nasal mucosa, because the same defect is present in the nose as is in the lung, but is far more accessible. And unlike the lung, is far more capable of being measured. So we could actually look at the transmembrane potential as a measure of chloride transport that could be corrected by this to really tell us, is this going to work? Unfortunately, it didn't show very consistent results. And so today there is no gene therapy for cystic fibrosis. And luckily, 20 years later, things have moved on in a very different direction. But I think in both circumstances, it turned out to be incredibly valuable to use a biomarker to help direct and make that go, no-go decision in the early pharmacology and pharmacodynamics. So, of course, the other areas would involve looking at that drug effect and further down the pathways in trying to give us some indication of real efficacy, and that's used certainly in our phase 2 setting. And that's been spoken to a fair amount here. And of course, the value that biomarkers bring there has been this idea that They reduce the heterogeneity to a great degree within a small population that you might want to study in a phase 2 and in multiple different dosing populations. And hence, a great advantage can be taken by using relevant biomarkers in that setting, and at the same time using safety biomarkers to give us early signals before we actually signals, for example, of kidney injury, rather than looking later on for actual clinical evidence of kidney injury, and being able to pick those things up early within your dosing regimens. And then we come to enrichment. And enrichment is really one of Bob Temple's favorite topics. Bob's out there, so he can always comment. But here, that's really the heart and soul, I think, as Mike was trying to say, and I'll underline, of what is precision medicine. And of course, in enrichment, we've already mentioned that you can have prognostic enrichment, in which you use a biomarker to identify when a patient is most likely to have an event, and therefore you can better detect your signal with that prognostic. But the truth is, I think most of us, when we talk about precision medicine, we're really focused on the predictive aspects of various different biomarkers, looking at those who are most likely to respond, or those who are most likely not to respond, or those who are most likely to have a safety problem if in fact they are treated. And finally, and only finally, I really say, we come to the biomarker that is likely to predict clinical benefit that we then call surrogate endpoints. Now, my point here a little bit is that the level of evidence for the use at each one of these different points and the utility for each of these is different, and the value is also different at each one of these different points. And biomarkers may not necessarily translate from one to the other, even for one product to another product, and therefore precision medicine actually has to be precise not only to the patients and the patient populations that these biomarkers relate to, but even to the products that will be used to them. And hence why, in the scheme of things, it's sometimes very much easier to try and look at biomarkers within a specific product development program under an IND for us than it is to qualify them for general use, because the burden of proof then becomes much larger. So I think we've talked a little bit about this use of predictive biomarkers being based on our knowledge of the biology, where it becomes clearly the most useful and where we, I think as Mike pointed out, we can really use them very early on to begin to narrow our population and increase our ability to identify the signals that we're looking for with much smaller and less costly efforts because of that reduction in the heterogeneity and even the very specific location of, say, a genetic mutation that we know that this particular drug is going to address. Mike then talked about this use in phase 2 of trying a more empiric approach in which we might search for a biomarker that can help separate subgroups. And, you know, that clearly is a bigger problem. So is the fact that I've run out of time. But I was actually trying to think of what was I going to say in all this time. You know, so to close up, I guess, you know, much has been made of the omics revolution in this setting of potential search for those biomarkers. And I think the point is maybe that we still have great promise, but not a whole lot of delivery yet today, that approach has not yet borne great fruit. But I do believe that it might be the way that we will be able to rescue drugs that, in fact, like Ivacaftor, Lumacaftor, identify a very small part of the population that's being studied, even in a phase 2, and allow you to then separate that responsive group from that non-responsive group. From a regulatory aspect, I think in this idea of enrichment, and I won't go on too much more, it's simply to say that one of the issues for us is to think about how to then label a product for its indication. And I know that Bob's going to take this up probably in his next session, but I think it is one of the key issues that we face around precision medicine and the use of predictive biomarkers. Thank you.
Thank you, Alice. Jumping step.
I guess I would lead excitement if I fell down the stairs. Wait here, wake everybody up. So thank you for inviting me. I wanted to give a perspective in terms of precision medicine in oncology. I think that this is very early time for precision medicine and oncology, but I think it is the right time to really consider this. In the last few years, there's been a lot of drugs that's been approved in oncology that are targeted therapies that has a molecular target, like EGFR mutations in non-small cell lung cancer and translocations with crizotinib. If you think about this, the question is, should we actually then, if any histology has this particular mutation or molecular abnormality, should that drug apply across all histology? Well, unfortunately, that does not apply for at least some of them. For BRAF mutations, as you can see with vemurafenib, for melanoma, there is actually a significant response. But then in colon cancer, they actually did not see the response that they had expected and seen with melanoma. And part of this is they found this, that there is a resistance mechanism that potentially led to the fact that you do not see that efficacy. So the question is then, how much do we need to really study? Do we need to study every single histology for every drug that comes along? NCI has, for the last 2 years, been developing a study called the NCI-MATCH, or Molecular Analysis for Therapy Choice. And the objective of this study is to try to, instead of use histology, is to use the molecular target for our patient selection. For this trial to really occur, it really takes a village. There are hundreds and hundreds of people involved for the last 2 years in trying to bring this about. For one thing, in order for us to be able to select to find a molecular abnormality that potentially is in 1% of cancer patients, you need to screen a large number of patients. And so we are planning to use the National Clinical Trial Network, which has over 2,400 sites across the United States, including NCORP, which are the community cancer centers where the patients are usually seen initially. Not only do you need to be able to screen large number of patients, you need to be able, if you are going to take a patient through a biopsy and screen them for this abnormality, you need to be able to have good options for them. So if they only have 1% chance of going on study, then the risk for them is much higher than if they have greater percentages. So we have been in conversation and working very closely with a large number of pharmaceutical companies who have been great to work with in coming forward and providing us both their information that they have, confidential information regarding biomarkers that may not be published yet, so that it allows us an opportunity to assess that, to assess if that would be a good biomarker for us both to put in our assay and to include into the trial. We actually started in 2013. At ASCO had a general meeting with a lot of pharmaceutical companies who have stepped forward and said they were interested in participating. Since then, we've held multiple conversations with the Agent and Gene Committee that's been set up to discuss with each company their agents and then to select the agents that we would like to put into MATCH trial. Currently, MATCH is set to go with 10 arms. One of the way MATCH is set up is you have a master protocol and then there are sub-protocol arms within it. We would like to be able to, under that one IND, be able to file different sub-protocol arms so that there's some flexibility in adding new arms as information becomes available. We hope that within opening of this trial in mid-August to be able to add 10 additional arms quickly after that, so that it allows patients, when they are being biopsied, to have a greater chance of actually having an actionable mutation. Along with the pharmaceutical partners and the sites, we actually also had a lot of experts at the front end to tell us about the landscape, the genomic landscape at this current time for precision medicine. And we have sub-protocol young investigators to help us in running this trial. We also have been working with the FDA, exploring potential regulatory concerns, both at the trial level, the trial design, as well as the assay. So this is just a schema in which patients, when they come on study, do require biopsy because we want to know their molecular profiling at the time before they start any particular treatment. If they have an actionable mutation, they will then be assigned under an informatic device called Matchbox to a study agent. If patients have stable disease of greater than 6 months or a PR/CR, then progress, we would like to biopsy those patients to understand the resistant mechanism that potentially have cropped up that led to the resistance to the treatment that they're on. If they have new actionable mutation under that second biopsy, we will consider them for treatment in a different arm. This trial is based on level of evidence so that there is no tumor board to assess in terms of what arm patients are going on. This is actually level evidence for selections of drugs. As you know, there's probably multiple agents sometimes for one particular target. We've used the level evidence to help us select in terms of which target or which agent we would like to go with first. However, as drugs are developed, their level evidence will go up, and we would like to, especially for those arms that are more frequent and are likely to fill up with the 31 to 35 patients, to be able to have another arm to kind of exchange out as we need to. In terms of variants that are selected for our assay, it is also based on level evidence as stated here. For level 1 is gene variants approved for selection of an approved drug. Level 2 is if it's an eligibility criteria for an ongoing clinical trial or there's N of 1 experience. And then level 3 is preclinical inference data. And the importance of this is that Matchbox uses this if a patient has more than one variant in terms of selection for which treatment they will go on initially. These are the first 10 arms that is set to activate. As you can tell, the prevalences are relatively low, so it's important we have multiple arms for patients. As we say here, we would like to be able to have about 30% if a patient gets biopsy that they will have a mutation that will allow them to enroll onto MATCH. ECOG Akron is our partner for this particular trial, and they will be the repository for the specimens that are collected on this trial. We have standardized as much of the process as we can in terms of the sample collection and shipping, as well as the assay itself, which is using an IM-Torrent personal genome machine for this with 143 genes being tested. The assay is developed by Dr. Mickey Williams at the Frederick National Laboratory, and the 4 sites selected there has been selected under RFA to run the trial, to run the assay for the MATCH trial. And these are just the other precision medicines that are currently being sponsored by NCI. The MATCH is one that is yet to open. Alchemist and LungMap, it has been open and is predominantly focused on lung cancer. MPACT is a trial that is randomized to look at different pathways, and it is in progress at this point. And then Exceptional Responder is a phenotype to genotype in which we want to know drugs that did not lead to approval, but yet you always have that 1 or 2 patients in phase 1 that responded. We would like to be able to get those samples and understand why they responded when large number of the patients on the trial did not. Thank you.
Well, thanks, Alice. That is the safer way.
This morning, you heard 2 comments in the opening. One from Janet Woodcock talking about applying modern tools, and from Mark on needing to understand patients. What I want to talk about is going to shift us in our direction on the panel a little bit as a perspective. I want to look at participant-centered trials using pervasive computing devices to capture real-time information and reveal the actual features of the disease, because we're interested in what would this do to pre-enrollment, what would it do to assessments, and what would it do to post-approval after drugs have been approved. Up till now, precision medicine has primarily been focused on defining subgroups. You've talked about in oncology and other areas, but in fact, it hasn't really focused on what I think is maybe the most important aspect for precision medicine not considered, which is the realities of fluctuations. The hypothesis I want to drive here is that better tracking of patients at baseline and understanding the fluctuations in daily changes in what their disease is feeling like, how it's manifesting, will allow more accurate measurements of interventions, especially for changes in the quality of their life. And I think this will fundamentally set how you will actually tell whether a drug is or is not working. Right now, we're sort of the beneficiaries of an enormous amount of genotypic data that's come in, and I and others have participated a lot in looking at subgroups and thinking about this, but we have not focused enough on the other element of precision medicine, which has to do with the phenotypic data. I'm going to argue that if you don't have rich phenotypic data, it's not going to do you much good to have all this subtyping on the genotypic. So we have to tip this balance back a little bit. The problem is that most patients are voyeurs to their disease. They don't really feel invested in what's happening to them or what's going on with how they could change, what they could do about their disease. And we treat people in trials, in fact, as we call it, subjects, almost in the medieval sense of the word. You're a subject. It's a one-way dialogue on data that's being extracted. Similarly, the ways that we measure changes in disease have gotten to be so regimented, insensitive, that in fact it's hard to actually build. It takes tens of thousands of individuals and you've got to follow them for years. It'd be nice if in fact there were better ways to look at ways that smartphones could actually give you information that was more sensitive, more objective, continuous. It could be engaging patients, not just around a particular hospital, et cetera. So the Robert Wood Johnson Foundation funded us at SAGE over the last 3 years to look at how do you do participant-centered research studies with feedback loops, and how do you make it so that the participant feels equal to the person who is doing the research? What type of information flow would that allow? And most importantly, how could you turn anecdotes, this is making me feel better, this is making, into signals, and how do you reflect that N of 1 from one person to another? You may have seen in March of this year, working with Apple, there was a series of iOS-based mHealth apps for building clinical studies that came out from Apple called ResearchKit. They were built to be a framework for clinical studies. They have consents, surveys, tasks, and passive data, and they've been put together in a way where you can borrow them and use them in ways that you want. It's open code that's hosted on GitHub for anyone to use and evolve. Apple doesn't see any of the data, and Sage BioNetworks, for these elements of it, has functioned as a neutral repository for that data. The particular app I want to review today is around Parkinson's disease. My guess is you're aware of this Empower app. It does things like taking tapping interval tests that allow you to look at that. It looks at just looking at the voice quality and what's going on in terms of actually hundreds of dimensions of data we can pull out of that. Put the phone in the pocket, gait and balance, we can collect things like that, just like a physician would. Spatial memory tests exist in there. And we built vertical stacks that go between the validated ways of looking at surveys through structured tasks and up through passive measures in order to tame those and make those highly effective and less obtrusive, lower friction. Within the first 3 months of doing this, we had over 15,000 consented individuals, 11,000 enrolled, and it was in fact the largest Parkinson's disease trial that has ever been assembled. But here's where it gets exciting. This is day-by-day fluctuations. I've just pulled up one for a day. This is 90 days. Look at the baseline change in that patient that, it, quote, has stage 3 Parkinson's disease. Look at the change in the tapping. Look also at the change in L-DOPA. L-DOPA is not having the same effect. If you're industry and you're trying to run a trial and you pick the wrong day for when you're testing that, it's not going to look the same as others. You need to have this real-time information in order to assess what's going on. But more exciting is that instead of having a singular measure, like is in an electronic medical record, number of taps, we can pull out dozens of dimensions of data, first-order features, and it's important for the following reason. Here's a 62-year-old man and a woman, 67. This is the change in their tapping. L-DOPA looks like it's working well in the man. In the woman, it's not having much an effect. Using machine learning, we built personalized classifiers, taking all of those features, built classifiers, and for the man, tapping is the predominant effect for the L-DOPA pathway. But for the woman, it's actually affecting her tremor. So you can actually build up personalized scorecards that allow you to follow it. But we think the most exciting thing that came was we asked the patients every day, or as often as they wanted, to say, what's making you better or worse? It wasn't the medication, it was sometimes sleep, but we have now actually over 20,000 of these responses. And what we find is it's what's happening to their mood, it's happening in their life, is giving this massive change in basically how their disease is manifesting. It's not the drug, it's what's going on in their life. If we had a way to look at real loads and reliefs that are going on in patients, that might be able to modulate the symptoms of the disease. Then you have the backdrop on which you could actually measure your own drug and what's happening there. So we've been going from diseases as syndromes to symptoms. We now actually think it should go to specific features. We think it's possible to build a scorecard that you give to a patient to move to the clinician and say, here's what's happening in my disease. I don't know what it means, but I want a dialogue with you, the physician. What does this feel like to have this disease? You can do things like that for Parkinson's disease. We're very interested in Alzheimer's and dementia. As you may know, Sage is working a lot in this. And there are 7 or 8 ways that you could follow dementia. Imagine if you had those ways of tracking symptoms at that level of sensors for dementia, how much easier it would do to do an Alzheimer's trial. So what we've been doing is using federated approaches for digital phenotyping. We think there should be open data, open source code on the devices, direct comparisons, and test what's the best way to follow episodic memory. Don't use a black box from a commercial company that won't tell you how it's doing the assay. Have a real way to accelerate the adoption of a standard that could be used for regulators. So we think these participant-centered data from apps have actually 2 fundamental benefits. One is it allows a better understanding of the disease, but it actually makes the patient feel much better to have that level of understanding. We've been working with different movement disorders, rheumatoid arthritis, anemia, melanoma, working with a number of industry partners to figure out how you could do that. And there's an article that came out this week that I wrote as an editorial that's in Science Translational Medicine. You can look at it, and here's the last slide. There are opportunities to use pervasive computing to impact clinical trials, and I think you can divide them into 3 places. Pre-enrollment baselines, so you know what the patient is actually doing before you give the drug. Second, new secondary endpoints and ways to distinguish the effect in order to show a regulator or a payer that actually the drug is doing something better or different. And third, when the drug is approved, imagine using pervasive computing devices as to track all the unexpected symptoms or issues that may come up in a drug in a way that you can't possibly afford to do in other ways. You could have groups of 10,000 patients being followed for what's happening to them and avoid seeing mistakes that come up and aren't recognized. Thanks very much.
Steve, thanks for your presentation as well. So we do have some time for questions and comments from those of you who are here, so I'd encourage you, as before, to come up to the microphones for any questions or comments on this important set of topics related to precision medicine. While waiting for people to come up, I'm going to go back. We covered a lot of ground in this session. I'm going to go back to where Michael started and focusing on, I guess you call it the typical, maybe it's the model approach of trying to develop some evidence on markers before you get to phase 3. And in the case studies we went through, that was certainly the payoff from that was evident. Could you all comment on the challenges of the alternative approaches and whether you see any possibility for going there? That is more marker evidence developed later in the process or even post-market. I wonder if that fits with some of the things that Stephen was just talking about too. Any thoughts?
I'll let Mike have first crack, and then I'll take a charge at it.
So just so I understand the question, so you're asking about generating this information in later stages of development rather than trying to get it early. Yeah, I mean, it's been a...
But you haven't seen that so often, and it's implying some real challenges with it.
The so-called prospective, retrospective approaches obviously would be very attractive because you could still enter a phase 3 confirmatory trial stage of development, but also do some learning about biomarkers in a very planned and structured way. And that would obviously help to meet that goal of getting a precision medicine and understanding in whom the drug is actually being effective. It becomes very challenging for procedural reasons, logistical, related to sample collection in a lot of ways. We find that globally, for example, DNA collection, the regulatory ethics boards have very highly different perspectives on that, and it's not possible to get voluntarily collected samples on all patients in a clinical trial. So when you have a subset of a clinical trial, it brings in statistical issues related to bias and other factors. So it's been a challenge, but obviously it would be clearly the best of all worlds kind of approach to developing precision medicines.
So a few thoughts around that, I guess. Where this has worked to date, and it's been popular. I think Mike was talking earlier about the rapid growth of targeted therapies, for example. We've gone from about 5% 10 years ago to as many as 45% of, depending on your accounting method, of recent approvals have been targeted. And if you look at breakthrough therapies, for example, about 80% of them carry a targeting strategy. So all of those were early, right? They all came into this usually with a very early focus. And that's clearly where the strategy's been the most successful to date. And I think that probably reflects the science. In Alzheimer's, the science related to amyloid has been pretty well worked on for many years. The issues in CF, also in terms of the genetic mutations and the type of mutations and the evolution of how they affect function, I think were very important. And then if we look in oncology and antivirals, where this has been really used in a very successful kind of way, it was the war on AIDS and the war on cancer that developed the molecular biology that really allowed us to understand this mechanistic, biologically understood approach to identify markers that, in fact, led us to these great successes today. And in almost none of these have we been able to do this retrospective examination to if you will, identify the new biomarker. Now, I don't think we should give up. I still think it's really important because I do think that there are many drugs in the past that have gotten buried, but maybe could have really worked in a subset, and where we could have identified that subset, or we could have done a better job around identifying the issues that limited the effectiveness from maybe their metabolic approaches, et cetera. So I think it's going to be important. We just haven't gotten there. But it does involve a longer course of acceptance. And that may be okay because it may have import beyond the regulatory issues. It may have to do with reimbursement. Ultimately, if you find that, yes, you had a drug that worked on 60% and therefore your phase 3 trial still was successful, albeit maybe the p-value wasn't out of the woods, but it was successful enough to get approved. But the margin of benefit was relatively small for the average. But there was a subset that you subsequently identified using a biomarker that came from phase 3, for example, where it was highly effective and had a much bigger margin of benefit. I think that information will play very importantly into the future of that drug for the sponsor and for patients in the choice of which patients will or won't receive that drug. So I do see that there is a real future for that.
Great, thanks. You guys are so polite.
Ben Shepherd from the Institute for Alternative Futures. One of the challenges in moving forward with the personalised medicine field can be developing a compelling value proposition or a business model where there can be concerns that you're sort of fragmenting the market and whether a one-size-fits-all blockbuster model approach provides more of a business sense than, say, focusing more on looking at how we can target and identify biomarkers. has the panel, whether we can provide sort of experiences or suggestions on how we can potentially tackle that environment so we can be very much patient-centric and to say that there is a good value proposition there, maybe just with this drug, but also across the portfolio of other ones that may come on board over the future years.
Rich?
I don't want to monopolize the microphone, but I guess I'm going to speak with my previous biopharm hat as opposed to my current regulatory hat. But, I think the companies that are thinking in that blockbuster, let's treat them all, and, if a few of them benefit, that's great, are going to become dinosaurs. That is, and I think most companies of today are recognizing that and are understanding that, in fact, what is important to the reimbursement world is this value. And if a drug can have 100% likelihood of benefiting and a significant margin of benefit, then that's a drug that's likely to be reimbursed. And that's, I think, what the wisdom for many companies is becoming.
Great presentations. Thank you. I have two questions. The first is related to the cases that were presented. These development programs were built on very strong foundational knowledge. And my question is about iteration. How do you build in contingencies for emerging knowledge in development? And that's even more challenging in regulation. So, we have our adaptive design guidance, and it probably talks more about what we're not comfortable with than what we are comfortable with in terms of adaptation. So I'd be curious to hear about that. And then the presentation that you gave, I noticed FDA is very interested in patient-focused drug development, as you know. I noticed what was not on there was construction of novel primary endpoints for drug approval. And I wonder if that was deliberately sort of left off because that's super futuristic and may never happen, or what your thoughts might be on that.
Any comments on the first question first, then we'll get to Stephen on the second?
So I guess as one of the two industry reps, I have no choice but to respond to that question. And I'll actually begin by kind of returning back to the previous question related to an appropriate, how you actually develop an appropriate business model for thinking about how to have these really customized medications for subgroups of the population. I'd say that our commitment is ultimately to allow access to all patients. It's just a question of making sure that the regimen that they're going to be delivered is going to be efficacious for those patients. And so, to that end, that business model, the way that would be structured is to continuously evaluate all of the additional data as they're generated. And not necessarily your question specifically was about adaptive design. I won't go into anything related to adaptive design, totally different trials in the space of CF as compared with oncology. But, where we would think about it is, continuous investment on the preclinical side in order to advance the pipeline so that we're able to provide access to more patients and iterating the clinical data that are being generated in order to further refine future plans, both from the standpoint of preclinical and clinical development.
Alice, you described for MATCH the use of potential adaptive arms, up to 10, I think that you said. Was that assisted by FDA guidance on the issue too, or?
You mean the additional 10 arms that are, no, these are, we have, initially we wanted to start with 20 arms partly because we wanted to be able to at least have a third of the patient when they're biopsy to have an option. But the timing of getting everything, it took a lot longer to get everything up and running as we anticipated. So but we have had multiple conversations with various members of the FDA regarding more the trial design, the master protocol everything's filed under one IND, and then CDRH regarding the assay. So,
any other comments on the first question? Then we'll go to Stephen.
So I thought that was a remarkable, refreshing question. I think that the dimensions, the features of disease that can now be captured have a great opportunity that we had assumed could fit as sort of secondary endpoints because they might be considered as something you could add on. The power of them as primary endpoints is obviously more impactful. I think that what's going to happen with many diseases that we're anticipating for Parkinson is the existing therapies may assume that these are the symptoms you're able to affect. If you have a more dimensional set of features that you're using to follow disease, it is easy to expect that actually a new drug might provide benefits that aren't along the same axis that the first drug is. And I think those are what you're referring to. And those are really powerful because they offer the patient something that the original set of drugs did not. And so I think
and Stephen, just to follow up, and Rich, do you have a comment? Go ahead.
No, I was just saying it strikes me that that's really important to do if you could in early phase. This is where it's a nice readout to begin to understand. One of the things you always want to do in phase 2 is identify what endpoints really does your drug impact, and how do you measure its benefit on the variety of possible endpoints that are available to you. And so this ability to look at multiple different areas in real time, I think, opens up great possibilities of trying to identify how you might best be able to approach phase 3.
Is there any kind of qualification process needed for that?
No, I assume it's going to be done under IND, and I think, it would be evaluated on what you found in your early...
If you lose, you lose.
Friends. Yeah, if you lose, you lose. But this increases your chance of winning rather than losing. Because, it's always been problematic in drug development to pick a single endpoint that, in a way, is unidimensional of a complex disease. And so the greater you can capture the possibilities of real benefit, I think your greater chance of success, ultimately.
On the early-late question, it's worth remembering that erlotinib was not anticipated to work in EGFR positive. That was done after the phase 3 study was complete. That's one reason we didn't limit the use to EGFR positive people. We're now seeing people looking at results of early studies and looking at groups defined by various SNP arrays to see whether they can identify a responder population or not. I wouldn't say any of those have been entirely successful yet, but we've seen that in especially the psychiatric area a lot, where the diseases probably aren't single gene diseases, they're probably multiple. And of course everybody knows about MammaPrint, which is used to try to identify people who are more or less likely to have metastatic disease from their breast cancer. So some of these obviously require that you have a study in hand before you can do these things. So I think that's going to go on too.
Thank you. I got time for 2 more quickly.
Okay, thanks. Marshall Hope, Genentech. This is a question for Dr. Chen, but others can weigh in as well. The NCI plans in oncology, they're quite impressive. How do you deal with tumor heterogeneity? Because you take biopsies, which is one specific location, and particularly if you have metastases, you may actually, of course, deal with numerous mutations in various spots of the body. So if you care to comment on that, thank you.
Yeah, I mean, in terms of, one, we're limited by areas and tumors that we could biopsy. So usually it would be the liver, skin, and so the question of tumor heterogeneity is an issue that I think right now it's a little difficult. We are looking in terms of how they assess the mutation. They're using percentages, but obviously I think technology as part of the adaptive is that technology as it improves, I think it offers us opportunity to continually evaluate this, how we want to assess the tumor. And we are looking as part of correlatives for MATCH to look at new ways to biopsy patients, new ways to assess those biopsies as well. But we are somewhat limited by technology of today.
Thanks. Michael?
Don't want to prolong things here, but there's a very interesting comparison between your presentation, Dr. Chen, and yours, Dr. Friend, of the new technology is here. it's a no-brainer that we need to use it in order to help drug development move forward. No question, there have been a lot of obstacles that you have to face, many of them unanticipated. What's the one thing you wish, perhaps from a regulatory, legal, administrative standpoint, you wish were easier and maybe could get things moving more quickly if we were able to overcome it?
I'm not sure regulatory was really what limited the moving forward for this trial, because we have, I think we had multiple conversations with the FDA, and they've been very interested, I think, and very helpful. So, and I think the one thing that we also have set in place is to try to use a central IRB for the network so that there is not this prolonged, because I think in the past, multi-center studies really has been delayed in each site having to open through their own IRB. So I don't, at this point, it actually, from the regulatory standpoint, I think it actually has
So what is it that's led to the delays and has us, not yet really open and launched?
I think part of it is really just having these multiple meetings. I guess, as we develop the studies, there's been more issues come up that we didn't anticipate regarding, like, when to inform patients of their results, having to get expertise in setting up, in terms of the assay, the validation of the assay across different sites. The informatic has taken some time in terms of Matchbox, in that once we set something in place, if we need to make any changes, it takes a little more time. So I think those has been more the things that have delayed somewhat.
Well, I mean, you mentioned, the multiple IRB issues. It also, seems like the patient selection is more challenging in your case than it is in Stephen's. As far as I know, the Apple phone doesn't yet do NGS, but so you need those centers and some kind of reliable process for getting the assay results to stratify your patients, right? So that makes it a bit more complicated.
We do that at each of our individual medical centers, though, with great regularity at this point in time.
Mm-hmm.
So, but so an unforeseen delay in getting that standardized.
Mm-hmm.
No, the assay is just that we wanted to use one assay, and it has to be validated, and then we had to show that it's reproducible across all 4 labs that are being tested. So I think each one of those, but also it's just as we've gone into this large trial involving multiple partners, some of the contracting issues with each partner, is kind of just little things. I don't know that's one big thing that delayed everything. It's just some unforeseen things and then some unanticipated. But most of it are just, I think we have worked through a lot of it, and having the mechanism and the expertise to be able to deal with it as it comes up. So.
Very good questions. Great discussion. I want to thank you all for an excellent panel. Thanks.